Hello students, in this video we will be going through the 2022 G.C.E level biology MCQ paper. Now on our last videos we went through the 2023 paper and the 2024 paper, and the link for those videos will be given below. Now let's go straight into the 2022 paper. So the first question of the 2022 paper was regarding lipids. Now this question, the first question always comes from Unit 1. It's a well-known fact, right? So on the recent year, the 2025 paper, which occurred a few weeks back, they asked about the nucleus and the nuclear pores and the nuclear membranes. And the year before that, they asked a question, very easy question. They asked about what was the basic functional and the structural unit of life. And before that, they asked a question regarding the lysosomes and peroxisomes. So you can sometimes expect a very easy question like what is the structural and the functional unit of life, but sometimes they might give questions from other cellular organelles, from proteins, carbohydrates, lipids. So this on 2022 they gave a question on lipids. Now let's go through the first answer. Or before that, what are lipids? Like carbohydrates, like proteins, lipids are also nutrients that are essential to the body. And when it comes to lipids, there are three main types of lipids, right? We have phospholipids, we have steroids, and we have fats, right? So here they are saying lipids are macromolecules consisting of carbon, hydrogen, and oxygen. Now, what is the other nutrition that consists of carbon, hydrogen, and oxygen? They are the carbohydrates. Like carbohydrates, lipids also contain carbon, hydrogen, and oxygen, which is partially correct. But are they macromolecules? The thing is, we can't consider lipids as macromolecules because they are not very long enough. Like if you take carbohydrates, specifically polysaccharides. Polysaccharides have very long branching chain. So we can consider them as macromolecules. But lipids, they don't have very long chains. So we can't consider them as macromolecules. So that statement is wrong. So second statement, each hydrocarbon chain of saturated fats contain one double bond. Now as I told, fats are one type of lipids, right? And fats are made up of glycerol and fatty acids. So this fatty acid, it can be saturated or unsaturated. So if the fatty acid is saturated, it means it is saturated with hydrogen atoms. And it doesn't have even a single double bond. But unsaturated fats, they have double bonds in between them. But the statement here is saying each hydrocarbon chain of fatty acids contain one double bond. As I told, saturated fats doesn't have even a single double bond. Why? Because they are saturated with hydrogen and they only have single bonds. Again, that statement is also wrong. And third statement, when fats are formed, glycerol and fatty acids are joined by hydrogen bonds. As I told, fats are made up of glycerol, which is an alcohol, and fatty acids. Now, where the name acids come from? It comes from carboxylic acids. Now, when alcohol and carboxylic acid, when they join together by ester bonds, not by hydrogen bonds, right? When they join together, they release a water molecule and join together by ester bonds. So that is also wrong. And fourth statement, hydrogen to oxygen ratio in lipids is higher than 2 to 1. Now, again, what is the nutrition that contain a H2O ratio of 2 to 1? Those are the carbohydrates. But do lipids contain a higher ratio of H2O? Yes, right. Why? Because, for example, let's take fats. Fats are made up of glycerol and fatty acids. As I told, fatty acids have a very long hydrocarbon chain, right, with only made up of carbon and hydrogen. So because of this long hydrocarbon chain, they do have a lot of hydrogen atoms. So the ratio is way more higher than 2 to 1, right? So that statement is the correct answer. Anyways, let's take a look at the final statement. Final statement says two phosphate groups are present in a phospholipid molecule. Now here you can see this is a molecule of the phospholipid that is present in the resource book. So here you can see the phospholipid is made up of two fatty acid chains, right? So here you can see one fatty acid chain and another fatty acid chain and one phosphate group, not two phosphate group. That statement is also wrong. So the correct answer is the fourth answer. So the second question is regarding a compound light microscope. So they are saying lenses reflect light to magnify the image of the specimen observed. Now, in a compound microscope, does the light is reflected? No, right? The light is usually refracted in the compound microscope to form the image, right? Light is, in other words, the light is bent. So how does a compound microscope works? So if you take a compound microscope, they have two lenses, right? They have the eyepiece lens and they have the objective lens. So here I have added a diagram how rays pass through the microscope in a compound microscope. So here you can see they have a eyepiece lens and they have a objective lens. So first the light from the object, here you can see the small red arrow. So the light from the object, they passes through the objective lens and they form an image. Now this image is a bit more bigger than the object, right? So this image is used to form a way more bigger image by the eyepiece lens, right? So let's go through the answers. So that's just a summary of the how the lens works. So this statement is wrong because lens refract the light to magnify the image. As you can see, refraction is taking place, not reflection. The light is bending here, right? You guys, you can see it, right? So second statement, resolution power is inversely proportional to the wavelength of light. So what is the resolution power? So the resolution power is the ability of a microscope to separately see two different points. Let's tell a microscope, let's take a telescope, right? If you take a telescope and you see a star, so in one telescope you can see two stars and in another telescope you can only see one star. But in the telescope that you can see two stars, there's a very high resolution power because they can distinguish 2 points separately. Same with the compound light microscope, they also have their own resolution power and it is inversely proportional to the wavelength of light. That statement is correct. And for this reason only we have the electron microscope. Why does the electron microscope has a higher magnification than the light microscope? Because of electrons. Electrons have a shorter wavelength. When they have a shorter wavelength, they have a high resolution power. So they can magnify the image way more better than a light microscope. So that statement is the correct answer. Resolution power is inversely proportional to the wavelength of light. Now let's go through the third statement. The image produced by the eyepiece lens is magnified by the objective lens. So here you can see image is produced by the objective lens first and that image is magnified by the eyepiece lens. So it is actually the other way around than they have mentioned in the statement. So that statement is also wrong. The fourth one, the maximum magnification is usually 600 times the actual size of the specimen. Now, in a light microscope, the maximum magnification is 1000 times the actual size of the specimen, not 600 times. That statement is also wrong. Finally, the resolution power is 0.2 millimeters. So in a compound light microscope, the resolution power is not 0.2 millimeters, it is 0.2 micrometers. So this is where you have to be really careful, the values and the units. Like you have to give more importance to the units. Most students, they get the values right. They know the value, they know the value for the cell size, the size of a virus, bacteria. They know the cell size, but where they go wrong are the units. They don't know the proper units. And in most questions, they actually give the right values. As you can see here, they have given the right value 0.2, but they have played with the values. They have changed the values. So that's what I say, always go to the values, because I think that's where they make the change, the usually it's the micrometers and nanometers and millimeters. So always be wary of that, always be careful where unit is used. So that's something to remember. So the second answer is the correct one. Now let's go through the third statement. The third question. A function common to both rough endoplasmic reticulum and the smooth endoplasmic reticulum is. Now we know rough endoplasmic reticulum and the smooth endoplasmic reticulum, they are both organelles that are present inside the cell, right? But they do have very distinct functions, different functions. So first let me think, synthesis of glycoproteins. So is synthesis of glycoproteins common to both rough ER and smooth ER? No, right? Because glycoproteins are synthesized by the rough endoplasmic reticulum. Right, that statement is incorrect. Second one, synthesis of phospholipids. Now, synthesizing phospholipids or any kind of lipids, it can be fats, steroids, they are all functions of the smooth endoplasmic reticulum, not the rough endoplasmic reticulum. So this is also not something that is common. The third one, metabolism of carbohydrates. Again, metabolism of carbohydrates also is a function of the smooth endoplasmic reticulum and the rough endoplasmic reticulum. And fourth statement, production of transport vesicles. Now we know in both rough endoplasmic reticulum and smooth endoplasmic reticulum, proteins are synthesized, right? They synthesize their own substances. Now these proteins and other molecules, they have to be released, sometimes to the Golgi apparatus for further modification and packaging, or maybe outside the cell. But in both rough endoplasmic reticulum and smooth endoplasmic reticulum, they are released in special vesicles called transport vesicles, where the substance or the protein or the molecule is packaged inside the vesicle and sent to their location that they are required. Most of the time, this location is the Golgi apparatus. And sometimes for like glycoproteins, what they do, they are sent into the cell membrane. So that is the correct answer. Finally, we have storage of calcium ions. Again, storing calcium ions is a function of the smooth endoplasmic reticulum. That is also wrong. So here you can see the functions of the rough endoplasmic reticulum and the smooth endoplasmic reticulum according to the resource book. In the rough endoplasmic reticulum, proteins, they are transport proteins synthesized by ribosomes. They synthesize glycoproteins and produce transport vesicles. And they also produce their own membrane, so we call them the membrane factory. Then smooth endoplasmic reticulum, they also produce transport vesicles, so that is the correct answer. But they also metabolize carbohydrates, synthesize lipids, and also stores calcium ions. Now let's go through the fourth question. Which of the following statements regarding subcellular components of eukaryotic cell is correct? So DNA and ribosomes are present in thylakoids. Now, what are thylakoids and where are they present? They are present in the chloroplast of the plant cell, right? It is in the thylakoids. The pigments are present that are involved in photosynthesis, and their DNA and ribosomes are present in the thylakoids. We know in the thylakoids, the photosystems are present, not the DNA and ribosomes. As you can see in this image of the chloroplast that has been given on the resource book, DNA and ribosomes are present on the stroma, which is the fluid-filled matrix in the chloroplast, not in the thylakoid membrane. Right? That statement is also wrong. The second statement, outer membrane of the mitochondrium contains Stokes particles. Now, as you can see, so this is a structure of the mitochondria. Mitochondria have an outer membrane and an inner membrane. And the inner membrane, they form cristae. As you can see, this structure, we call it cristae. And Stokes particles are present in the cristae of the mitochondria, not in the outer membrane. That statement is also wrong. Third one, glyoxysomes have enzymes required to convert fatty acids to glycolipids. We know glyoxysomes, they are specialized peroxisomes, right? And they do not convert fatty acids to glycolipids, they convert fatty acids into sugars. That is also incorrect. Fourth one, Golgi apparatus manufactures cellulose. So this statement is correct. Why? Because Golgi apparatus manufactures both cellulose and non-cellulose components such as pectin. Right? So that statement is actually correct. So always remember, they don't manufacture the cellulose entirely. They manufacture the subcomponents of the cellulose, which can be transported to the cell membrane where cellulose is made. Now let's go through the fifth statement anyways. Chromatin is embedded in the nuclear lamina, which is made up of protein. What is chromatin? Chromatin is the DNA protein complex. In other words, DNA histone complex. And they are saying it is embedded in the nuclear lamina. But chromatin is embedded in the nuclear matrix, not the nuclear lamina, which is made up of proteins. So that statement is wrong because chromatin is embedded in the nuclear matrix, as I told, not in the nuclear lamina. Okay, let's go through the fifth question. Which of the following statements regarding enzymes is correct? Activators affect the function of enzymes by binding to active sites through covalent bonds. Now here they are talking about allosteric activators and inhibitors, right? We have learned in allosteric regulations about these activators and inhibitors. So there are some activators affect the function of active sites through covalent bonds. So activators, we know they don't bind to the active site. They bind to sites other than the active site via non-covalent interactions, which means they don't bind permanently, right? They bind reversibly. Change the shape of the enzyme in a way the substrate can bind to the active site. So that statement is wrong. Second one, shape of the active site of enzymes changes due to temperatures higher than the optimum temperature. So this statement is actually correct. What happens at high temperatures? The bonds that are present in the enzymes, such as the hydrogen bonds and all the other bonds, what happens? They start to get broken. Because of this, the shape of the active site changes. So that statement is actually correct. So the third statement says many competitive inhibitors bind to active site of the enzyme irreversibly and change their shape. Now competitive enzyme inhibitors, they bind to the active site, which is partially correct. But do they bind irreversibly? No, right? They bind reversibly and change their shape, right? Because they are reversible inhibitors. That statement is also wrong. Fourth one, toxin binds to enzymes reversibly through covalent bonds. Now always remember, whenever something binds through covalent bonds, they don't bind reversibly. They bind irreversibly or permanently. So that statement is also wrong because toxins bind permanently through covalent bonds. Final answer, coenzymes are proteinase components which are permanently or temporarily bound to enzymes. Now, what are coenzymes? Coenzymes are organic cofactors. And organic cofactors, they are non-proteinous components. And the other part is actually correct. They bind permanently or temporarily. Which are permanently or temporarily bound to enzymes. but they are not proteinous components, they are non-proteinous components. Now let's go through the sixth question. So the electron transport chain. So what is the electron transport chain? So it is the final stage of cellular respiration, right? The first stage we have glycolysis, then we have pyruvate oxidation, citric acid cycle, and finally in the electron transport chain, we generate ATP. So they are saying it's located in the matrix of mitochondria. So the electron transport chain is present in the inner membrane of the mitochondria, not in the matrix of mitochondria. That statement is wrong. Second statement generates two molecules of ATP on average due to oxidation of one NADH molecule. Oxidation of one NADH molecule usually produces around 2.5 ATP, not 2 ATP. Okay, that statement is also wrong. So third statement uses molecule oxygen has the first electron acceptor. Now we know in the electron transport chain, molecule oxygen is the final electron acceptor. Okay, not the first electron acceptor, because finally the electrons are accepted by oxygen in the electron transport chain. But if you are talking about the first electron acceptors, they are the NADH molecules and the FADH2 molecules, right? Because they are the first molecules that are accepting electrons in glycolysis, pyruvate oxidation, and in the citric acid cycle. So this statement is also wrong. Fourth statement generally generates one molecule of ATP on average due to oxidation of one FADH2 molecule. One FADH2 molecule produces around 1.5 ATP, not 1 ATP. That is also wrong. Fifth one is composed of protein and non-protein molecules. That statement is actually correct, because electron transport chain has proteins and non-protein molecules such as coenzymes and cofactors. So that is the correct answer. Okay. The seventh question is again regarding cellular respiration. So they are asking 4 events of aerobic respiration of a glucose molecule are as follows. We know respiration of glucose molecule, aerobic respiration has 4 parts, right? Actually 3 parts: glycolysis, pyruvate oxidation, and citric acid cycle. And finally, we have the electron transport chain. So let's go through the process. So let's mark it. So glycolysis occurs first, right? Then we have pyruvate oxidation, right? What happens in glycolysis? Glucose is converted to pyruvate. Then pyruvate oxidation, where the pyruvate is converted to acetyl-CoA. Then in the citric acid cycle, the acetyl-CoA enters the citric acid cycle and generates more FADH and NADH. Then finally, in the electron transport chain, ATP is generated. So first statement, they are saying release of 2 carbon molecules by decarboxylation. Now, in which stage carbon dioxide molecules are released? So on 2 stages, carbon dioxide molecules are released. That is during pyruvate oxidation and the citric acid cycle, right? So it's on 2 and 3 during pyruvate oxidation. When pyruvate is converted to acetyl-CoA, carbon dioxide is released, and also during the citric acid cycle, carbon dioxide is released. Then B, oxidation of NADH and FADH2. Now, don't get confused. Oxidation of NADH2 and NADH and FADH2 means they are losing electrons. So they are losing electrons in the electron transport chain, which is the final stage, right? They lose the electrons to produce ATP. And for the third one, the C1 and D1, oxidative phosphorylation and substrate level phosphorylation. Now, what is oxidative phosphorylation? So oxidative phosphorylation is producing ATP using the electron transport chain. Right? When we produce ATP using the electron transport chain using the NADH and FADH2, actually using the electrons in the NADH and FADH2, we call it the oxidative phosphorylation. And substrate level phosphorylation are the ATP we produce using other substrate. I hope you guys remember that we produce ATP during glycolysis, right? We produce 4 molecules of ATP in glycolysis, and we produce 2 molecules of ATP in the citric acid cycle. Now these ATPs are produced using substrate phosphorylation. So oxidative phosphorylation, it occurs in the final stage, ATP producing using NADH and FADH2, and substrate phosphorylation occurs in the first stage and the third stage. Now let's write the correct sequence. So here we have subcellular phosphorylation. It occurs in glycolysis. D, and second one B, oxidation of NADH and FADH. So that statement is actually wrong, right? So the D answer cannot be the first one, because oxidation of NADH and FADH2, which occurs in B, should come finally, right? Because ATP is generated in the electron transport chain. So let's go in this way. So let's go through from the second answer, right? So release of carbon dioxide molecules by decarboxylation. So it occurs in the second stage, pyrite oxidation, right? So A. So it's got to be either first one or the second answer. Then what happens? Then we can go through substrate level phosphorylation, right? Which occurs in the third stage. So second stage, release of 2 carbon dioxide molecules by decarboxylation, which occurs in pyrite oxidation. Then substrate level phosphorylation, which occurs in the citric acid cycle. So D answer is the second one, AD. Then B and C, which is oxidation of NADH4 and oxidative phosphorylation, which occurs in the electron transport chain. So this is the correct answer. Right? As you can see, A, which is released into carbon dioxide molecules, it occurs in pyrite oxidation. Then we have D, which is substrate level phosphorylation, which occurs in the citric acid cycle. And finally, we have B and C, which occurs in the electron transport chain. So which is going in the correct order, right? Without involving glycolysis in this. So that is the correct answer. Now let's go through the 8th question. The 3 eras of the Phanerozoic eon and 5 events are given below. Select the answer indicates the correct era-event combination. So we know we have 3 eons, right? Sorry, 4 eons. We have Hadean, Archean, and Proterozoic and Phanerozoic eons. And we have 3 eras in the Phanerozoic eon. We have Paleozoic, Mesozoic, and the Cenozoic eras. So the first era of the eon Phanerozoic is Paleozoic. So it is in this era the life started to evolve. The life in the sea, the fishes, the insects, the formation of vascular plants, it all occurred in the Paleozoic era. And here they have given 5 events, and in this, origin of reptiles and dominance of amphibians occurred in the Paleozoic era. So let's write it. So it is the origin of reptiles and dominance of amphibians. Then second one we have Mesozoic era. So it is in the Mesozoic era mammals started to originate, right? And also gymnosperm starts to dominate. That is R, right? Dominance of gymnosperms. Finally, in the Cenozoic era, what happened? Radiation of birds, which is the 5th answer. Sorry, the 5th event. So, and what else occurred in the Cenozoic era? The evolution of the mankind, right? The genus Homo. So in genus Homo, we have Homo sapiens, which are humans, right? And now we are still living in the Cenozoic era. Anyways, let's go through all the answers. So, wait, let me, okay. So A should either go with Q and S. So in all the 5 answers, it is going with S or Q. so we can't eliminate any answers yet. And the B, Mesozoic, B should go with P or either R. And in the fourth answer, you can see B is going with S, which is actually wrong. So we can eliminate that answer. So let's eliminate it. Then third one, Cenozoic, it should go with T, right? So C should only go with T. So we can eliminate the second answer and the fourth answer, because in the second answer, C is going with R, which is dominance of gymnosperms, which is wrong. Then on third answer, C is going with Q, origin of reptiles, that is also wrong. So it's got to be either the first answer or the fifth answer. So first answer, A is going with Q again, so which is correct, and B is going with Q, which is wrong, because in the Mesozoic era, origin of reptiles didn't take place. Origin of reptiles occurred in the Paleozoic era, so we can eliminate the fifth answer also. So the correct answer is the first one. Now let's go through the ninth question. Which of the following is considered an artificial group in the classification of organisms? At a little bit of context here, Robert H. Whittaker, right, the person what he did, he classified organisms based on their cellular structures, their reproductive methods, then what are their cell walls made up of. So using all this, he classified the organisms. So he classified them into bacteria, protista, fungi, plant and animalia, not arthropoda. Arthropoda is a phylum in kingdom Animalia, so we can eliminate that answer, right? So what is the artificial group here? We have two artificial groups, right? We have protista and we have virus. Virus is also another artificial group, but they have not included viruses here. So protista is the artificial group, right? So any organism that didn't belong to bacteria or fungi or plant, what they did, they added them to the group of protista. And protista, they don't have a single common ancestor, right? If you take bacteria, fungi, plant, they all have a single common ancestor, but protista doesn't have a single common ancestor. So they are the artificial group. So second one is the correct answer. So 10th question, select the pair which is most likely to have the highest number of common characteristics. So they always, in the recent papers, they always asked a question on the taxonomic hierarchy, the common characteristics from domain to species. So the domain is on the highest order in the hierarchy, which means they have the least common characteristics. But if you take organisms in the same species, they have the highest common characteristics. So as we go down the order, the number of common characteristics increases. Right, first we have bacteria and archaebacteria. Now bacteria and archaebacteria, they belong to completely two different domains, which is the highest in the order, which means they have the least number of common characteristics. So that is the wrong answer. The second one, we have Annelida and Nematoda. Now we know Annelida and Nematoda, they belong to the same kingdom, but they are different phylum, right? Phylum Annelida and phylum Nematoda. But they both belong to kingdom Animalia. So they are also somewhat high in the order, right? So they have very less common characteristics. That statement is also wrong. Then third one, we have bats and whales. Now if you take bats and whales, they both belong to the same kingdom Animalia. They both belong to the same phylum, which is Chordata, and they both belong to the same class, which is the mammals, so Mammalia. So for now, they have the highest number of common characteristics. So why? Because class is lower in the table than phylum, kingdom, and domain, right? So this can be the right answer. But anyways, let's go through the fourth and the fifth answer, and let's see if there is something more lower than in class. Then we have birds and reptiles again. Same kingdom, same phylum, which is a phylum Chordata, but birds belong to class Aves and reptiles belong to Reptilia. So they belong to the same phylum, which is higher than class, so it cannot be the correct answer. Then finally we have Lycophyta and Pterophyta. Lycophyta and Pterophyta, they both belong to the same kingdom, which is Plantae, but they are different phylum. So the correct answer is the third one, bats and whales. Why? Because they belong to the same class and they are the lowest in the order, right? So it is the third answer. Now let's go through the 11th question. Which of the following statements regarding seedless vesicular plants are correct? Now when it comes to seedless vesicular plants, we have two types, right? We have pterophytes and we have lycophytes, right? Now they are asking, they are saying all club mosses are homosporous, some pterophytes are homosporous, some lycophytes are heterosporous. Now, what does homosporous and heterosporous means? Homosporous means they have only one kind of sporangium and they only produce one type of spore, right? But heterosporous, they have two types of spores. They have the microspore and the megaspore. The microspore, they develop into the male gametophyte, and the megaspore, they develop into the female gametophyte. So under lycophytes, we have club mosses, right? And we have spike mosses. And pterophytes, they are all homosporous, right? Remember, they only produce one type of spore, so they are homosporous. But lycophytes, they can be either homosporous or heterosporous. So club mosses are homosporous, and spike mosses, they are heterosporous. They produce two type of spores. They produce the megaspore and the microspore. Now let's go through the answers. All club mosses are homosporous. That statement is correct because club mosses belongs to lycophyta and they are homosporous. Second one, some pterophytes are homosporous. As I told, all pterophytes are homosporous. Not a single one of them is heterosporous. So that statement is wrong. Third one, some lycophytes are heterosporous, which is again correct because spike mosses, they are heterosporous. So the correct answer is the A and C, which is the third answer. Now let's go through the 12th question. Which of the following features is present only in one class of phylum Chordata? Now in phylum Chordata, we have different classes, right? First, write down all the classes. First, we have class Condystites. Right, so let's write down. Right, class. Then we have Mammalia, right? That is also another class. Then we have Reptilia. Then we have Apes. Then we have Osteichthyes. How do I spell it? Then finally we have Amphibia. Okay. Now, the first statement they are saying ectothermy. So what is ectothermy? Changing their body temperature according to the external temperature. So what are the classes that has the feature of ectothermy? So that is the amphibia and reptilia. So they are not a common feature, right? So let's write it down. So that is the wrong answer, right? Then teeth. So in the resource book, they have clearly mentioned mammals have differentiated teeth, right? So class Mammalia, right? So only class mammalia has been mentioned on the resource book. Let's keep it like that for now, and let's go through the four-chambered heart. Resource book also mentions mammals have four-chambered heart, right? And also apes also has four-chambered heart. Is this mentioned in the resource book? Yes, in the unit 5, in the circulatory system of humans, it is mentioned that apes also have a four-chambered heart, just like mammals. So we can't consider them only having one class. Sorry, consider them. So consider four-chambered heart has only one feature in a single class because it is present in two classes. Then we have color vision. Now color vision is present in apes, right? Which are the birds. And also we know, which is not mentioned in the book, is mammals also contain color vision. If we take humans also have color visions, right? We have cones in our eyes that detect pigments. Sorry, that detect different colors. Then finally, we have the cycloid scales. Now, cycloid scales, they are only present in uh, osteichthyes, right? Which are the fishes. Now, it's got to be either the fifth answer or the second answer, but teeth is also present in osteichthyes. Why? Because birds has teeth, right? Sorry, apes. Sorry, not birds. Sorry, again, it's fishes, right? Fishes have teeth, which are the osteichthyes. So the correct answer is only the fifth one because cycloid scales are the only feature that is present in osteichthyes, right? Other all features are present in more than one class. The correct answer is the fifth one. Now let's go through the 13th question. Meristematic cells. Now what are meristematic cells? They are undifferentiated cells that can divide into differentiated cells when required in the plant cells. The first statement, they contribute to both primary growth and secondary growth. We know when it comes to meristematic cells, we have three types, right? We have apical meristems, lateral meristems, and intercalary meristems. And apical meristems, they are involved in primary growth, and lateral meristems they are involved in the secondary growth. Now primary growth in increase in length, and secondary growth increase in the circumference of the plant, increase in the diameter of the stem. So that statement is actually correct. Second one, meristematic cells undergo both mitotic and the meiotic divisions. Now meristematic cells only undergo mitotic cell divisions. It is the reproductive cells that undergo meiotic cell divisions. That statement is wrong. Third one may be either spherical or elongated. We know one of the characteristic features of meristematic cells is that they are isodiametric, which means they are roughly spherical. But they can also be elongated. They become elongated in the zone of elongation, right? In the roots, the meristematic cells undergo elongation, but their default shape is spherical. So that statement is wrong. Fourth one are present only in root tips and shoot tips. As I told, meristematic cells, we have three types, right? We have apical meristems, lateral meristems, and intercalary meristems. So apical meristems, they are present in root and shoot tips, but lateral meristems, we have coke cambium, uh, which increases the circumference of the stem. Then we also have intercalary meristem, which is present in the base of the leaves, which is involved in the repair of the plant. That statement is also wrong. Fifth, uncontained nucleus shifted to a side due to presence of a large central vacuole. Now, another characteristic of meristematic cell is that they have a large central nucleus. So this statement is wrong, and this is a feature of the parenchyma cells. So correct answer is the first one. Now let's go through the 14th question. Which of the following contribute to the opening of stomata? We know during photosynthesis, the stomata opens and closes, and this is controlled by the guard cells. So let's take a look at the summary of the opening of stomata. First, what happens? Accumulation of potassium takes place. Accumulation of potassium causes the movement of water inside the guard cells due to low water potential by osmosis. This increasing water potential, what happens? They start to increase the thermal pressure in the guard cell, and the guard cell starts to expand. But they expand non-uniformly, where the inner walls move away from each other and open the stomata. So first statement, increase in turg of guard cells, which is correct, right? When water enters inside the guard cells, the turgor pressure increases, right? This turgor pressure causes the guard cells to expand and open the stomata. Production of abscisic acid, we have learned abscisic acid plays a role in the closing of the stomata, not in the opening of the stomata. During drought, when there is very less water, it closes the stomata, it doesn't open it. So that statement is wrong. Third one, flow of water from guard cells into stomatal cavity. Again, this occurs to close the stomata. To open the stomata, water has to enter the guard cells. Final answer, accumulation of K plus in guard cells. So this is the first event that takes place in the opening of the stomata. The potassium ions, they enter the guard cells, which causes the accumulation of water inside the guard cell. So correct answer is A and D. So let's go through the 15th question. Passive movement of water and hydrophilic solids across membrane with the help of transport proteins is called. Now, whenever transport proteins are involved, it is facilitated diffusion. Why? Because hydrophilic solids or polar molecules, they can't pass through the cell membrane, right? So they have to use even water can't pass through the cell membrane. So they have to use transport proteins, right? So using transport proteins, solutes move from high concentration to low concentration without using any energy. So the answer is facilitated diffusion. As you can see, here they have given us 5 types of transport methods, and all these are passive movement of water. It doesn't require any energy. If you take diffusion, is a movement of substances from high concentration to low concentration through the cell membrane, but it doesn't use any energy. Then we have osmosis, which is the movement of water from high concentration to low concentration uh, without using energy. Then we have imbibition, which is the absorption of water by hydrophilic substances. Then we have finally have bulk flow, which is the movement of substances in large amount using the pressure gradient. Again, it also doesn't require any energy. So 16th question, nitrogen and boron can be absorbed into plants respectively as. So nitrogen can be absorbed as either NO3 minus or NH4 plus, right? So it's gotta be either the first 3 answers, right? We can eliminate the 4th answer and 5th one because not as NO2 minus or NO. Yeah, only the 4th answer we can eliminate it because in 5th answer also they are using NO3 minus. And boron, they are absorbed into plant has H2BO3, right? So first one is the correct answer, right? If you take nitrogen and boron, nitrogen, they are macro molecules that are required by the plant. So macro elements actually, right? Macro elements that are required by the plant, and boron, they are micro elements that are required by the plant. So first one is the correct answer. Let's go through the 17th question. Which of the structures of an or angiosperm is diploid? Right. So here we are talking about the flowering plants, and we are talking about whether it is diploid or haploid. So if you take a megaspore is formed by a megaspore mother cell, right? So it is a haploid because megaspore mother cell, they are diploid and they undergo meiosis to form the megaspore. So this is it is haploid. Then the microspore is also haploid, right? The microspore develops into the male gametophyte and the megaspore develops in the female gametophyte. So obviously they have haploid number of chromosomes. The third one, we have the ovule. Now the ovule is the cell that gives rise to the megaspore. So ovule should be diploid, right? Because the megaspore is haploid. So ovule should be diploid because the ovule undergoes meiosis to produce the megaspore. So that statement is the correct one. Fourth one, we have the pollen grain. Again, the pollen grain contains the male gametophyte. Again, that is also haploid. Then we have the embryo sac. So embryo sac in plant is also is the female gametophyte and it is also haploid. So third one is the correct answer. So 18th question. Plant hormones that regulate cell division, promote leaf senescence and enhance apical dominance are respectively. So regulate cell division, there is only one hormone, or in other words we call them plant regulators. So there is only one hormone that is involved in cell division, that is cytokinins. So it's got to be either the fourth answer or the fifth one. Now, when it comes to promoting leaf senescence, what is senescence? Senescence is the aging of the plant. So here this is specifically asking about leaf senescence. So if you take abscisic acid, they are specifically involved in leaf senescence, but ethylene also involved in the senescence of the plant generally, but specifically it is the abscisic acid when it comes to the leaf. So it is the fifth answer. And auxins, they enhance apical dominance. So what is apical dominance? Inhibiting the growth of the side but allowing the plant to grow only taller, higher in length. So the answer is the fifth one. Now let's go through the 19th question. Select the correct answer based on the following two statements. A function of connective tissue is providing support. You know, when it comes to tissues, we have four types. We have epithelial tissue, we have connective tissue, we have nervous tissue, and we have the muscle tissue. Now, connective tissue provides support and strength to all the other kinds of tissues. Right? For example, one of the well-known connective tissue is the bone. Right? Bone gives support to the body, and majority of tissues that are found in the body are connective tissues. This present all over the body, and these tissues they can contain three types of fibers. They can have reticular fibers, they can have collagen fibers, or they can have elastic fibers. So here they are saying reticular fibers provide strength to connective tissues. It is actually the function of the collagen fibers. So collagen fibers are the ones usually give strength to the connective tissues, not the reticular fibers. So the correct statement is the third one. A is correct, but B is incorrect. Yes, because first answer they are saying B contributes to A, it doesn't. And second answer they are correct, but B does not contribute to A. B is totally wrong. So it is the third answer. And the final question, both synthesis of vitamin B12, recovery of ions, and fermentation of undigested material take place respectively in. Okay, so synthesis of vitamin B12 occurs in the large intestine, specifically in the colon of the large intestine. Right, so in the resource book they haven't mentioned vitamin B12, but they have generally mentioned vitamin B complexes. So vitamin B12 comes under it, so it is the colon. Then recovery of ions, it occurs in the small intestine, and fermentation of undigested material takes place in the cecum of the large intestine, which is the first part of the large intestine. So it is the fifth answer. Okay. Now, on our next video, we'll be going through the next 20 MCQs from the 2022 paper. Thank you and see you guys.